Mitosis on Friday, but we still, I don't think, need to meet tomorrow. Okay, so no lab tomorrow, we'll meet on Friday to finish up the course. You have until 11:55 p.m. tomorrow evening to do the GFP lab, or lab report, which is the Angel quiz. And if not today or tomorrow, then on Friday I need to also get a physical paper copy of your mitotic index, which you can turn in as a group, which I strongly encourage you to turn in as a group. Okay, so all that is happening in the next couple of days. Today we need to finish up mitosis. So whenever we met last, which I guess was last Friday, we looked at the cell cycle and saw that we divided it into first two sort of large phases: interphase, not dividing, and mitosis, dividing. But then we subdivided interphase into G1S and G2 and saw that different things were happening in those different sort of substages. Okay, so you want to know what's happening in which of those substages. And we saw that different types of cells go through this process of mitosis at different rates, some vastly different rates. Okay, intestinal epithelial cells twice a day, you and liver cells only once a year, so there is a very, very broad spectrum here. And then we looked a little bit at what controls this process, and we saw that it was a combination of proteins called cyclins, whose concentration cycles, hence the name, and different cyclins peak at different times along the cell cycle. So this one is showing us the M cyclin, so it peaks during M phase, whereas others will peak during the other phases. But we saw that sort of the real work, in a sense, is actually done not by the cyclins, but by cyclin-dependent kinases that are there all the time. So these cyclin-dependent kinases are in your cell, or in our cells all the time, but they don't work unless the appropriate cyclin is at the right concentration. The two hook up together, the CDK gets turned on, all sorts of target proteins get phosphorylated by those CDKs, and those phosphorylated proteins are then able to control the cell cycle. So we looked at two examples, the transition between G1 and S, and then G2 into M, but all the transitions sort of work in essentially the same way. And we just got started with the actual process of mitosis. Looking again at a little bit at basic chromosome structure. After the S phase and after the chromosomes have copied themselves, the two copies, usually called sister chromatids, are held together by this protein called cohesin. You'll see that again in anaphase. And then the phosphorylation that's happening between the G2 to M transition, one of the targets, one of the many targets of the M cyclin CDK is this protein here called condensin, which starts to condense the chromosome. Histone H1 is another important target that will also contribute to condensing the chromosome into that visible structure that we looked at last week in lab. So that's happening sort of inside the nucleus during prophase. I think we've gotten to prophase. We saw the nucleolus disappears because ribosome production is shut down during cell division. Meanwhile, out in the cytoplasm, we're going to ignore all the other organelles pretty much and just focus on the centrosomes that have already been copied, right? That happened in G1. The two copies start moving apart to opposite sides or opposite poles of the cell, and in between them, the microtubules are developing that we call the spindle that will, as we'll see in a couple minutes, start connecting to the chromosomes and start moving them around. So I think that was as far as we've gotten to prophase. Okay, so far so good. How are we feeling about the cell cycle and cyclins and CDKs and at least prophase of mitosis? all sorts of extra time to look at this, I'm sure. Oakley. Okay, so from prophase we go into prometaphase. The hallmark event is the breakdown of the nuclear envelope. It's the hallmark event of prometaphase. We've actually studied this before. If you remember back in when we were looking at nuclear structure, I actually I showed you the same picture. So worth quickly showing you this again. Remember the inner membrane is coated with all those lamins, right, a type of intermediate filament. And we saw that when you phosphorylate those lamins, that causes the nuclear envelope to break down into little pieces. Okay, so that's going to be another target of the mitotic cyclin CDK complex are these lamins. So they get phosphorylated, the nuclear envelope breaks down, and basically that's going to then release all the chromosomes, or dump them in a sense, into the cytoplasm so that they can start getting picked up by microtubules. Okay, so if we go back to this picture, right? So you have the mitotic spindle here. So some of those microtubules will now start attaching to the chromosomes at those centromeres, at those constriction points. So we have something like this. Look at one chromosome. Okay, so here's a centrosome over here, the other one's over there. Now I'm growing all of these microtubules, and some of them are going to start attaching to the chromosomes at this constriction point called the centromere. But as people studied that interaction between the microtubules and the centromere, found that it's sort of not a direct connection. There are proteins, I'll go in green here, that sort of coat the centromere sections of chromosomes. These proteins are called kinetochore proteins. And it's really to those proteins that the microtubules are going to attach. But since they're coating the centrosome, we usually at least sometimes jump over them and just say it connects to the centrosome, or not centrosome, I'm sorry, the centromere. So be careful with all these centro words. But really, these proteins coat the centromere, and then that's really what these microtubules start attaching to. Now, in most of the animations that we're going to look at, it's going to appear that only a single microtubule attaches from either side. That is not accurate. It makes sense to draw it that way in an animation, but as you can see here, really clearly in this electron micrograph, there are a whole bunch of microtubules that attach to either kinetochore. Because in reality, one microtubule wouldn't be strong enough to pull the chromosomes around. So there are actually lots of them. There's a better picture. Yeah, there's a good picture here. Okay, so here's parasystem chromatids. The green is representing the kinetochore proteins. And again, multiple microtubules are attaching to those kinetochore proteins on either side. And since these microtubules connect to kinetochores, these particular microtubules that actually connect to the chromosomes are referred to as kinetochore microtubules. Because as we'll see in a few minutes, not all of these spindle microtubules do this. A lot of them do, because you need a lot of them to move the chromosomes around, but not 100% of the microtubules will do this, and we'll need to distinguish between the ones that do and the ones that don't. So the ones that do connect to chromosomes are called kinetochore microtubules, again, because they're connecting to these kinetochore proteins. So how to make sense? Okay. So those are the two big things that happen during prometaphase. Nuclear envelope breaks down because of the phosphorylation of the lamins, and then that basically gives access to the chromosomes, or gives access to the microtubules to the chromosomes. Okay, so they're going to start, at least some of them, are going to start connecting to chromosomes via these kinetochore proteins that are attached to the centromeres of the chromosomes. So there's a decent amount of vocabulary here that you need to associate with this. Okay. Does this general idea make sense? Connecting now the microtubules to the chromosomes. Silence means yes. Okay, so that's prometaphase. Metaphase occurs when all of the chromosomes are moved by these kinetochore microtubules to the center of the cell. And that's what we're showing here. So this particular example has 4 chromosomes. So this could be a fruit fly cell. They have 4 chromosomes. And each of them is lined up at the center of the cell. And that center is usually referred to as the metaphase plate. Chromosomes align at the center. They're going to line up individually. So we have 4 chromosomes here. Each one lines up individually. And I'm stressing that here because on Friday, when we study meiosis, that's not going to happen. Okay? So that's going to be a hallmark difference between meiosis and mitosis. So mitosis, chromosomes line up individually down the metaphase plate, down the center of the cell. At least usually. Okay? For most body cells, they line up right down the middle. Again, we'll see an exception to that on Friday as well. When some cells go through meiosis, they don't line up quite so evenly at the center of the cell. But we'll worry about that on Friday. Okay, so for today, for mitosis, they always line up individually at the metaphase plate, pretty much dead center in the cell. But notice then how this is probably going to work. Right? If we line up each chromosome individually, and each chromosome is composed of 2 identical copies, and one copy of each chromosome is attached to microtubules from that pole, and the other copy, presumably identical copy of each chromosome, is attached to microtubules from that pole, and kind of all we have to do is now pull the 2 identical copies apart, and each cell is going to get 1 of each chromosome, which pretty much is the whole point of mitosis, right? When we do carbon copies or Xerox copies of the cell. So that's about what's going to happen next, but are we okay with metaphase? Does that kind of make sense? So we're lining up individually. I know I'm really stressing that because that's going to be our big contrast for Friday. Making sense? Okay, so we start pulling those copies apart during anaphase. So here's sort of the setup. So we've already looked at then, in this particular slide here, the blue microtubules are the ones that are attached to the kinetochores, so they're the kinetochore microtubules. In this particular picture, the red or the pink, depending on how you see that, are microtubules that don't attach to chromosomes, but they sort of overlap with each other. They're either called interpolar microtubules or non-kinetochore microtubules. I'm used to seeing them called non-kinetochore microtubules, but your book calls them interpolar microtubules. These are microtubules that don't attach to chromosomes, but overlap with each other. And these little dots here are representing motor proteins. Okay, we studied those before, remember? So there are going to be motor proteins involved, well, actually with both populations, but especially with these. And as we'll see in a second, both of these populations of microtubules are going to be important for anaphase. Technically, there is a third population, these green ones that are called aster microtubules. We don't usually pay too much attention to them, but I'll mention them briefly here. These sort of come out the other way and kind of anchor the centrosomes in place is their basic function. So in reality, we have three populations of microtubules. Okay? Aster ones sort of anchor the centrosomes in place. The kinetochore microtubules attach to the chromosomes. The non-kinetochore or the interpolar microtubules don't, but they sort of interact with each other, and we'll see what effect that has in just a moment. So this is sort of the setup for anaphase, but we're still kind of at metaphase because they're all still lined up here in the center. I just wanted to talk about these different populations of microtubules first. Do the three make sense? Three populations? Okay, so what's going to happen? Well, the first thing that needs to happen, if we're going to pull apart the two copies, remember the two copies are essentially glued together by that cohesin protein. So the first thing we want to do, if we want to pull those two copies apart, we're going to break down that cohesin, that glue. That is the job of the anaphase promoting complex, the APC. That is yet another target of the M-cyclin MCDK complex, is to activate the anaphase promoting complex, which in turn activates this proteolytic enzyme, whose job is to break down all of that cohesin in between every single chromosome. So humans have 46 chromosomes. So this happens down the length of all 46 chromosomes seemingly simultaneously. When I show you a time-lapse film at the end, you'll see that all of the chromosomes seem to split simultaneously. How that really works and how that's coordinated is still not well understood. Now, whether it's a different, there are 46 different copies of this and they all somehow attach to a chromosome and they all work at the same time, or whether it's some other mechanism is not well understood, but it seems really clear no matter how many chromosomes there are, 4 in fruit flies, or 46 in us, or 70-something in dogs, or 16 in yeast, however many there are, all the chromosomes split at the same time. But again, how exactly that works, we don't know. But it has something to do with breaking down this cohesin seemingly simultaneously between the chromatids of each chromosome. So we keep linking all this back to that M-cyclin MCDK complex that's pushing the cell through mitosis at this point. Okay, so the anaphase promoting complex gets activated. That activates this enzyme, and probably multiple copies of that enzyme break all the cohesin, and now the two copies, the two chromosomes, are separated, and now they start being pulled towards opposite sides or opposite poles of the cell by these kinetochore microtubules. That event is known as anaphase A. Okay, so anaphase A is traditional anaphase. Chromatids are separated by breaking down all that cohesin. They start to be pulled apart by these kinetochore microtubules. Okay. And how does that happen? It's probably a combination of this treadmilling effect. So remember microtubules and the treadmilling, right? Individual subunits fall off the minus end, so that's probably part of it. But it's also very clear that there are motor proteins attached here at the end of the kinetochore microtubules and these kinetochore proteins. There are motor proteins there as well that are sort of actively walking along the chromosomes along the microtubules as they're shortening. So it's probably there are sort of two motors or two engines that drive this part of it that pull the individual chromatids apart. So all of that is anaphase A. The breaking down of the cohesin and the movement of the chromatids is known as anaphase A. Okay, so that's all about the kinetochore microtubules. Well, if there's an anaphase A, there's probably an anaphase B, and that has to do with those non-kinetochore microtubules, or these interpolar microtubules, as our book calls them. At the same time that the kinetochore microtubules are shortening, and thus pulling the chromatids, the non-kinetochore microtubules are getting longer. Right? Remember, microtubules are very dynamic structures. Some can shorten, some can lengthen all at the same time. So while the kinetochore ones attached to the chromatids are shrinking, and that's what's pulling the chromatids apart, these non-kinetochore proteins get longer, and they're pushing against each other, these proteins in here. And what that is basically doing is also helping to separate the chromosomes. It's really elongating the cell at this point. So chromatids are moving, the cell is getting longer and thinner, so that's helping to push the chromosomes apart as well. So anaphase A and anaphase B sort of work together to separate chromatids. Anaphase A is sort of the more direct part of that because the kinetochore microtubules are directly moving these chromatids, but the cell is getting elongated by these interpolar, non-kinetochore microtubules. So that's what's known as anaphase B. But they're both going on simultaneously. We just sort of separate them into two things in order to study it more clearly. But both of these events are happening simultaneously. So far so good? Makes sense? So we're almost there. So now we have separated a copy of each chromosome, and presumably two identical copies. One's going that way, one's going that way. And then the final phase of mitosis is telophase. Telo means last or final, so it's the final phase. Telophase is essentially the reverse of prophase and prometaphase. So the nuclear envelope broke down during telophase, it reforms. And actually not just one, but two nuclei, right? Because now we have two sets of chromosomes, one over here, one over here. So we get nuclear envelopes reforming around now both sets of chromosomes. And that is triggered by the dephosphorylation of those lamin proteins. So remember, when we phosphorylate them, the nuclear envelope breaks down. When we pull the phosphates off those lamins, they somehow, and this is not completely clear how all this happens either, but by pulling off the phosphate groups on those lamins, that's the trigger, at least, for the nuclear envelope to reform around each set of chromosomes. But there's still a lot we don't understand about how all that kind of works. But regardless of the lack of details, the nuclear envelopes do reform. So that's sort of the reverse of prometaphase. And then sort of the reverse of prophase, the chromosomes start to decondense because we're also going to start pulling off the phosphate groups off of condensin and off of histone H1, which will let the chromosomes uncoil. And almost immediately, the cell resumes its making of ribosomes, so the nucleoli reappear. So like I said, it's very much the reverse of prophase and prometaphase. And that, technically, is mitosis. Because remember, mitosis is actually, not simply, but it's just the division of the nucleus. Right? So we have achieved that. We started out with one nucleus. Now at the end of telophase, we have two identical, in this case, mitosis, identical nuclei. So mitosis, technically, is over. Right? Because we've divided the nucleus. Okay. Right, so let's pause here. How are we feeling about anaphase and telophase? Is this making sense? Okay. So while mitosis is over, almost always, but not always, but most of the time, it is followed immediately by the division of the rest of the cell, which is cytokinesis. And in reality, in most cell types, cytokinesis overlaps with telophase. But as I mentioned on Friday, they are separable events. There are chemicals that you can throw at the cell to allow it just to complete mitosis and not go through cytokinesis. Probably vice versa as well, that you can get it to go through cytokinesis without dividing the nucleus. So they are separable events, but if you're just watching this occur naturally, they kind of happen simultaneously, or cytokinesis and telophase usually happens somewhat simultaneously. So in animal cells, which is what we're going to focus on, if you look at the cell from the outside, what you see is the development of what's called a cleavage furrow. The cell starts to sort of pinch in from its center. So we're back to the center of the cell, because that's where this usually happens. And you start seeing the cell membrane sort of start pinching in. So we go from sort of a rounded, or in this case, since anaphase B has happened, sort of an elongated cell, which then starts to pinch in at the center. Okay, so that's the cleavage furrow here. And eventually, it keeps pinching in and pinching in until the cells are separated. So of course, as people were viewing this, they wanted to know what was causing this. And the answer to that is a ring of actin and myosin filaments right around the center of the cell, called the contractile ring. And much like the drawstrings on your hoodies, if you pull on those, what happens to the hoodie? It gets smaller, right? Same thing if you're wearing sweatpants. If you pull on the drawstrings, the waist of your sweatpants gets smaller. Same kind of idea here. So as these actin and myosin filaments sort of pull on each other, it pulls the membrane in until it pinches apart. So it's a nice little system for separating animal cells. Plant cells work very, very differently. We're not going to worry about plant cells for the exam, although one of the animations that I'll show you does show what happens, but I would like you to focus on the contractile ring of animal cells. So like I said, it's going to pinch in, again, usually right down the center, and eventually separate the two cells. So again, you can stop that by throwing in drugs that mess up the actin filaments, for example, and then this won't happen, and you'll have one cell with two nuclei, for example. And that, in short, is mitosis. So let's start looking at some animations and put some of this in motion. Oh, let's see.
The cytoplasm of a cell in late interphase contains two centrosomes, each of which may contain a pair of centrioles. In the nucleus, the chromosomes have been replicated during S phase, but are still dispersed in the form of chromatin. One or more nucleoli are present. During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of two identical sister chromatids joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle. During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole. At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles. Anaphase begins when the two centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, the spindle microtubules not attached to chromosomes lengthen, pushing the two poles farther apart and elongating the cell. During telophase, nuclear envelopes form around the identical sets of chromosomes at the two poles of the cell. The chromosomes uncoil and nucleoli appear in the two new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the two daughter cells. In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
Okay, so you don't have to worry about the animal cell part. Okay. Kind of everything in one shorter film.
Cell division consists of two processes, mitosis and cytokinesis. Mitosis, division of the nucleus and its chromosomes, is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is followed by cytokinesis, when the cytoplasm splits to form two separate daughter cells.
That's everything very quickly.
soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Okay, so that's all wonderful animations, which you should take a look at. All of these are up on Angel. But again, lest you think we make all this up, a couple of what I think are really cool time-lapse movies of actual cells. So here's the nucleus. Here's the nuclear envelope. Here's the beginnings of chromosomes. So they're condensing. So we're somewhere probably in prophase of mitosis here. And then the nucleus is gone. So now we're in prometaphase. And you can't really see the microtubules, but you can see their effects, right? Because the chromosomes are now moving all around. And the cell monitors this situation and won't start anaphase until all of the chromosomes are right in the center. So that little straggler one, everybody else is waiting for that one to come back in. And then all at once, they are all separated and moved apart. So there's anaphase A and B. Here's telophase and cytokinesis. Nuclear envelopes are reforming, chromosomes are decondensing. We have cytokinesis going on out here, which is pretty cool. Same thing here. Here's the nucleus. Okay, with the chromosomes. So again, I can see them, so we're somewhere in prophase. Here's the rest of the cell. It's kind of the perimeter of the cell here. Still there, still there, still there, still there, still there. Now it's gone. So now we're in prometaphase. Microtubules are attaching. They're moving all the chromosomes to the center. We're not there yet. Again, the cell can monitor this. And it doesn't move into the next phase until all of them are at the center. Split simultaneously there. So there's anaphase A, the cell's getting elongated, anaphase B, cytokinesis is occurring, telophase is happening, and we go from one cell with one nucleus to two cells, each with presumably an identical genetic nucleus. And that's going to be sort of the setup for Friday. Mitosis, the goal of mitosis is to give you genetically identical cells, right? So when your skin cells divide, your liver cells, or your muscle cells, or whatever cells you want to talk about divide, they're dividing into two identical copies, right? That's the goal of mitosis. The goal of meiosis, that we're going to look at on Friday, is not to have genetically identical cells, right? The purpose of meiosis is to create genetic diversity within gametes. So there are two big ideas, genetic diversity, and instead of, in the case of humans, two cells with 46 chromosomes like we see here, gametes, sperm and egg, are going to have half that number. So that when a sperm gets together with an egg, your new person still has 46 chromosomes. So those are the sort of two big ideas for meiosis. Is we're going to have half the number of chromosomes, and we're going to have non-identical sperm and egg cells that will promote genetic diversity. So that's what we're going to look at on Friday. So all of these animations are up on Angel, so I would encourage you to use those along with your notes on study mitosis. Again, the reminders, I'll make them again. We will meet on Friday to do meiosis and finish up the course. We will not meet for lab tomorrow. However, you need to get, if you have not done already, you need to get onto Angel no later than 11:55 p.m. tomorrow. Actually, you need to do it before that because it's going to shut off at 11:55 p.m. tomorrow night. The GFP assessment on Angel, and at least by Friday, if not today or tomorrow, I need your mitotic index lab report. Okay, so you have until Friday to get that to me. Okay, I'll take it now if you have it. If you haven't picked up quizzes, they're up here as well, so feel free to do that. Okay, so we will meet again on Friday to finish up the course with meiosis.